last session, that is a panel discussion. So I invite all eminent speakers for this session to assemble here for the panel discussion on this immunotherapy and biological session. Dr. Taruna Madan, Professor Abhay Pandey, and Dr. Kausik Chattopadhyay, and Dr. Ravi.
I would like to invite Professor Ravi Chandiran for the panel discussion. Please take this.
Yeah, please. Yeah. So the topic is very vast, immunotherapy and biologicals, and the recent pandemic taught us the preparedness for the platform technologies in perspective to have the indigenous technology to fight with such infectious disease. I think in the panel, lot of efforts has been done by the India, and lot of technologies has been developed. So I think we should discuss what are the future infectious disease we should be the technology ready. So I invite comments from the each panel ki how our technology readiness should go hand to hand to fight with this such infectious disease. So Dr. Ravi.
I don't know why Sharma put me here still. I try to understand, and I wish to make it very clear that see, the antibodies and the vaccines are very, very important, and the utility in our country is that very huge. What it is happening currently, it is not very sufficient to attend the requirement. So the challenges, demand, and what we have, this is a very, careful to attend, careful to understand also. This is what we were trying to, understand. I was talking with Ravi Mishra a couple of times with his director also, how the NIPER and the IMTech can go together to develop that the requirement of antibody drug conjugate, whatever the currently it is used by our people and for management of the healthcare. The basic problem what I could realize, starting from that any of the gene, it is synthesizing the gene. Whether in India, indigenously, we are synthesizing the gene, synthesizing facility, how many facility we have. So we are outsourcing from that the different lab. Someone is doing of that the gene synthesis, and then we are cloning, and then antigen productivity it comes, the antibody it is happening, we are testing in that the reality of that, the paper aspect. How we are going for that, the productivity aspect, how we are going to motivate the entrepreneurship in the country, so which can serve that the basic requirement, the challenges of the society. These are the things I have in my mind. I am not expert in this that area of antibody or the vaccine, but still Sharma put me in that the certain things. Why you are sitting here? You go and sit and ask that certain. Still, I am happy that if I understand, I am really very happy. Happy in the sense. See, the young people, we are here to really respond. So, what's happening? The happening in the sense, more than 5 decades, these antibodies are in the market. We could not able to do it. I can say that we are in the zero. So why? Why such things? We could not able to do it. Only that here and there in Naipar Mohali, our Panda is doing, and the Girish Sagani is very much with that the part to mentoring the Panda. This is a wonderful aspect. We are very happy. But still, whether we could able to attend that the societal requirement, we should very seriously think about that, how we are going to energize our next generation in the area of antibody drug conjugate, vaccine, where very, very important in that affordability. This is very important. Again, I say that the affordability is all available. Materials are available. Whether we are fit into that to purchase this material and correct our problem, these are the question I have. Whatever you think that, you think on this, these are the requirement it is there. As a director, as a sniper, these many snipers are there. So you all attend this. We are very happy to attend the problem and energize our young people to learn and to become the startup and entrepreneur in the future, at least another 2-3 years period. If we start now, with a couple of years, we could understand that they will be energized to face the society in the future pandemic, any kind of the problem. So this is what I stop here. Thank you, Chair.
Yeah. So, thank you, Dr. Ravi. And now, views of the Dr. Taruna.
Thank you, sir. Thank you for the opportunity. I come from a, very distinct combination I have, that I did my graduation and post-graduation in pharmaceutics, and then I moved to PhD in a combined program of CSIR and College of Pharmacy. Now we know it as a University for Pharmaceutical Sciences and Research. And I was an anomaly actually in those days. Not many of my colleagues and friends were coming into basic research. And I chose that because I thought that I have that bent of analytical mind, that I can, do the experimentation and analyze whether it is going to be having some application. So the translational research that we know now as was not known in those days, but I had thought of that I want to discover something new which is needed for the society. And that's how I moved to CSIR, and we tried to develop a diagnostic kit, immunodiagnostic kit. That was my foundation. And then, of course, I moved on understanding basic research and then applying it to the human health. We have done experiments with clinical cohorts of women suffering from preeclampsia, and we have discovered biomarkers that are of relevance. We have, first thing was to go for candidate approach. When you know this protein is important or this molecule is important, and you go for that. But now, these days are omics approaches, that we have big data and analysis of big data using bioinformatic tools, and then coming to those authentic targets, because candidate gene approach has a bias. I love that molecule because I had worked on that molecule, so I want to project that is the best molecule. But omics gives you that ability to have molecules without a bias And my thinking is that we are really moving faster on the path, as Dr. Ravi said, that India has lagged behind, and that's what DBT Secretary Dr. Rajesh Gokhale also mentioned in my recently attended microphysiological systems, an EMBO workshop was being held at CCMB Hyderabad, where they have projected that microphysiological systems are an answer to validate the molecules that you have using human organoids, human organ systems on the chip, rather than using animals. So the tagline of the Atal Incubation Center at CCMB, which is called CPHMS, is humans are not 70 kg rats. So they want that human systems should be used, and a Boston team has actually developed such systems, and we are almost there, that these systems would be validating our molecule. And Rajesh Gokhale actually said that India has always, not cashed upon the upcoming technologies, and we should be fast enough to, cash on these technologies. We should adapt them, adopt them, and then we use them. Thank you, sir.
Very rightly said, and thank you. And Dr. Chattopadhyay, your views on the future generation of the biologics and early adoption of the technologies on the immunotherapy and antibody conjugates and biologicals, vaccines?
I'm probably complete an odd man out to make comment on these because I was trained as a chemist, then turned into a biochemist.
But your molecular mechanism and the odd crosstalk will lead to the such development of such candidates, definitely.
Right, I mean, I should say maybe the field is unfortunate or I'm fortunate, I don't know. Because for example, during my postdoctoral training, I mean, of course, during my postdoc research I was doing crystallography, protein crystallography. But my target proteins were all these co-stimulatory molecules. I mean, I was trying to crystallize and co-crystallize protein like ICOS, ICOS ligand, which are nowadays a remarkable marker for positive co-stimulation. And during that time, Jean Allison, who got Nobel Prize a few years back for immunotherapeutic approach of co-stimulatory molecules. So I always try to look at things from a different perspective. I like to see in that a different direction. That of course we need basic understanding molecular approaches to understand the mechanism, what is going on inside. At the same time, applicability of the processes, that we should always be thinking. Like when the pandemic started and there was complete lockdown in the initial days, and so all the students, they left labs were completely shut down. I was thinking what to do during that time. Then, that time, okay, there were some previous manuscripts to be written up, some grant application, God knows when to be submitted, but I was doing my background work. But very interestingly, Professor Balram gave the keynote lecture yesterday here, and also he gave a colloquium at ISER Mohali. And that time he was talking about how he spent his time during lockdown. He was searching for the person who discovered coronavirus during 1968. So, and then of course, I mean, during this corona period, what was amazing, I'm sure the feeling goes same for everyone, that we have been hearing vaccine development approaches since my childhood. I mean, when I did my PhD in NICED, National Institute of Cholera and Enteric Disease, which is an ICMR institute, and that's why I had, of course, that time I was doing protein folding, completely odd man out in that institute. But I knew like people are trying for vaccine development, this thing, that thing. And I was always thinking like, why so much effort? It takes time, takes years. But the corona period showed us that a pathogen became, or we became appreciative of the fact that a new pathogen can come, emerge, and vaccine can be developed. Not only that, vaccine can be implemented, and I don't know, maybe I'm surviving now because I got 2 dosage of vaccine and a booster dose. So I think ultimately all this collective approach, basic, translational, whatever it is, we have to keep on working and we can only try.
Yeah. So early adoption of the technology platform and early to discuss with the industry partners. If we have the technology, definitely we need to discuss early with the commercial partner so that can be goes to the public. And so Dr. Ipsita.
Yeah, thank you. So I think it was a very enriching panel and enriching discussion. And what we probably need to appreciate is how we design our experiments, and we have been doing that for a very long time. But to incorporate those factors which will help us, like we always say that, okay, so this is what I can do in an academic lab and that's it. So even if I cannot do it, for somebody else to take it forward, there must be some parameters that should be built into the experimental design so that they can take it forward, that I reach somewhere and then I am told that no, you cannot take it forward because of 1, 2, 3, 4, 5, 6 reasons. So if that can be built into the curriculum somewhere, maybe at the postgraduate level or as a pre-doctoral course, so then I think it helps to channelize our efforts much better, and this consonance that we are talking of between basic research as well as the applications of it will also become more meaningful. So that is, I think, what I would like to say.
Yeah. So you reminded that some challenges like early points to consider for the development of technologies. Like in the morning we discussing about HIS-tag. Majority of the academic labs uses the HIS-tag for the purification purpose. When you go to the regulatory, they don't like it. They hate it. You have to remove it. So coming back to the removal of the HIS-tag, it is again 1 year job. So I think academia has to also consider in the mind what is approvable. Otherwise, all the efforts which has gone to the proof of concept development, which will go in the vain, and in the pandemic type situation, we cannot afford that. So such type of things we also consider early. What are the regulatory requirement in the complete process, because it is a difficult. Biotech is a difficult. It is not easy. You have to be reproducible at every step. So now coming to taking the views.
Yeah, thank you. Yeah, I agree with you. Regulatory detail need to be, our students or coming generation need to be exposed to these details, because as a scientist, for example, our biotechnology students may not know these aspects when they will go into the development of product or process. And this is a very high time when the demand of such actions are needed from all over the nation. We need to have a central framework whereby our students need to be told about various aspects of regulatory affairs which they are going to meet when they will grow as a scientist and take their product into the field. So yes, I agree. Histidine tag is one of the examples. There are many thousands and thousands of steps which need to be told to the student, and which also need to be simplified from the government point of view to make the process relatively simplified. I will not say easy. Drug development is not easy, and so that some product can come out of it. Thank you.
So Dr. Ravi, yeah.
Yeah, thank you. So I'll start with Dr. Ravi Chandran's comment, because we have been discussing also on this aspect. First thing, with respect to monoclonal antibodies. That's good that we are, word pharmacy, we are pharma, but now it's time for biopharma. And that's where, you need pharma and biotechnology to come together. Otherwise, it will not be never possible. And these biopharmaceuticals are having a lot of issues, particularly affordability. I will take an example of anti-cancer monoclonal antibodies. As Dr. Ravichandra was mentioning that the problem, where is the problem? The thing is, we have to really go in a structured manner. There are 4 or 5 problems in this domain which need to be solved. First, and very important, is the media. Being a part of few committees in BIREC, we have funded so many industries for developing affordable media for cell culture. So that's where academia need to play a role, because all these media are proprietary media. Huge 1 liter media costs 50,000. How you will make that? And the lab working, having less funding, they cannot really do that. So the media component is not a rocket science, and we have seen that so many firms in Pune and other, they come up with good comparable data with the Lonja and Thermo. So that's the one thing where we need to really focus, and government also giving a lot of emphasis on that. So designing affordable media which can reach up to with this internationally recognized media, and they have monopoly in that. So, and second thing is the cell line. There are just 3-4 cell lines used for the monoclonal antibody. CHO is the one, 75% of the MAB are based on the CHO cell line. And there are multiple variants of it. But again, those variants are patented. Thermo, Lonja. They have their own patent and coming with a huge royalty. One cell line which you are using, CHO K1, you can get from 80cc 1 lakh. And you take CHO-S from Thermo, it is 40 lakhs, along with the licensing agreement. And that's nothing. That's we in the fundamental science we do every day. You look at their vector. It's a dual promoter vector having the SV40 and the DHFR and the DHFR knockout in the host. And that's fine. It's we use those things, but those kind of effort, although all the time this effort is not fruitful, you don't get a very good publication out of it. But those kind of things we do routinely. What I learned during this COVID time, looking at the what kind of dependence we have for the initial material. Take an example of Covishield. That's a viral vector. We always doing that. It's not a rocket science. Express spike protein. We are doing every day for our research, but that is not our priority. So those things we need to be to prioritize it. One. Third thing is looking at the industrial compatibility, as you are mentioning just now, that engaging industry in the very beginning is very important. Otherwise, we will be going the dead end. And that's most of the program is going like that. Second thing is that we cannot do everything. We have to be very selective. And for that, we have to look at, very simple is that, because all the monoclonal antibodies are patent protected. And fortunately, these patents are going to expire by 2030. Majority of innovator molecules are going to be expired by 2030. What we do in our group and our lab, we look for the patent which is going to expire in the next 5 years or next 3 years, and to start the work on them, not on the very fancy molecule which otherwise you will end up nothing. It will be patent protected. You will not be able to do it. Even you will not get the sequence. Getting the sequence is the most important challenge, and you have to do multiple places. Find out the right monoclonal antibody sequence. So that strategy, if we go, irrespective of what kind of publication and scientific thing, if you want to develop a technology. So looking at the patent and which is going to expire, we can start on that, because that is the need actually. And I was part of one of the committee that constituted by Prime Minister and headed by DGCSIR 2020. And the agenda was why all the early development of the molecule in pharma is happening outside of India, and they are coming and asking for clinical trial here. And this is a big challenge, and people are aware of it. Why these early development, they do outsource outside, bring the molecule, ask to ECGI, give me the permission to phase 1, or even give me exemption for phase 1. I would like to go to phase 2. So where we can pitch in, why this is happening, and that is not, again I will say, the rocket science. So we have so much rich science and technology institutions that, so those kind of, that is one of the point which was highlighted.
Yeah.
In that context, actually, to develop the early molecule, we got a program supported by BIRAC. So after that, they have now decided to make 2 facilities for the cell bank, the GMP compatible cell banks. So one, I am leading the program in IMTech. It's a BIRAC supported program for mammalian cell NCCS. So they have selected 2 institutions in 2020 and a huge funding. It's not, funding is not limitation. They have given fund to make a 2,000 square feet GMP lab, all sort of analytical things. The thing is that you have to bring the, globally recognized cell bank technology to India to facilitate our newcomers. Because for a new industry, importing cell line from ATCC, another thing is the big challenge. They will not listen even to them for the, in terms of agreement and all. So government wants to bring those expression system host in India, as many as we can, and provide a platform to innovators to take the things from here and develop their program. At the same time, the mandate is to connect Indian scientists who are working in this domain and bring on that platform. Let's say if he's working on some molecule, he has a very interesting expression system, but otherwise it's not useful for him, it's just his academic use, and it's sitting in the fridge. That may be useful for someone else. Yeah. So those kind of networking, so we are now starting those things in that, so things are moving in positive direction, but a lot of things to be done. Yes. Yeah, thank you.
Thank you, Ravi. So, taking from you, one of the point you said that ki biopharmaceuticals, and pharmaceuticals we grown up such a huge way, and we are one of the largest exporter of the ANDA based products. But still, in the biopharmaceuticals, we are not doing enough. Now, there is a concept called, it is an old concept, but I think it is in a renewal process, antibody drug conjugates. Recently that drug called trastuzumab has been launched. So we need to innovate or ignite our young mind how to club the pharmaceutical agent with the antibody, and which is the antibody suitable for the which chemical agent. Mainly the application lies with the oncology. So I think in the PhD program or such programs and higher education program, we need to consider such type of projects where we can develop the affordable biopharmaceuticals for the Make in India process. Otherwise, like cost of the one therapy, like trastuzumab is 60 to 70,000, which is not affordable by the Indian, like majority of population. So I think in biosimilar line, we have done good job. I think the significant cost reduction has been done by the Indian companies, and a lot of innovation has done in the biosimilar area. But now, novel biologicals and biopharmaceuticals, we need to do a lot. Yeah. Any audience? Like?
Companies, since you are
technically, like in the manufacturing, we need to have the